So welcome once again to SBT 214, Biomembranes and Enzymology. Today we're continuing with biomembrane composition. And we want to first of all understand what biomembrane is. And if you take a cell, for instance, we take a cell, the cell has a cell membrane. That is the outer, permeable, selectively permeable covering of the cell. And even within the cell, you have some organelles that also have some membranes around them. So a biomembrane simply is a selectively permeable barrier which separates the interior of a cell and also even of organelles from their external environment. So you have, the cell membrane creating that barrier around the whole cell so that it will regulate the transport of substances across it from outside of the cell into the cell and from inside of the cell to the outside of the cell. Similarly, when you take the organelles inside of the cell, so for instance, if you take the nucleus is having the nuclear membrane around it. That's also a biomembrane. And when you take organelles such as the mitochondrion is also having a membrane around it. So mitochondrion is even having two membranes around it. So each of these nuclei, each of these organelles which are inside of this eukaryotic cell will be having such a biomembrane around it. Of course, for prokaryotic cells, you see that you don't have a distinct nucleus, but rather you have the nuclear material not defined inside of the cell. But if you take a eukaryotic cell, as you see that all those other organelles, in addition to the nucleus, all have some membranes around them, so that a separate compartment of the interior environment of the organelle is separate from the cytosol, the liquid part of the cytoplasm. Now, we want to continue to take a look at what biomembranes are composed of. When we say biomembranes, what is the composition of these biomembranes? We did look at the different kinds of lipid membranes the last time. In our last lecture, we did look at structures that can be formed due to the properties of the lipids, so that we saw micelles can form. We saw that you could have some liposome can form. All of these, structures which we looked at in the last lecture was due to the amphipathic nature of the lipids, having a hydrophobic head and having a hydrophilic head and a hydrophobic tail, so that you can have these lipids form a bilayer. So as you see here, the composition of biomembranes consists of mostly phospholipids, which are arranged in such a way that there's a phospholipid. It forms a phospholipid bilayer, and inside of this bilayer, you have other structures that are embedded, like some proteins. So you see some channel proteins. You see some glycolipids. So that is carbohydrates with some lipids in a complex there, forming. Generally, these glycolipids will be acting as some receptors on the cell for cell recognition, for signal transduction between cells, you do have some of these glycolipids. The same way you have glycoproteins, also complexes of carbohydrates with protein. So as you can see, there's a protein embedded in the cell membrane, and then there is some carbohydrate which is attached on that protein. Even these proteins, you can see there are different kinds of them. We'll come to actually in the next lecture, we will look at the membrane proteins, specifically looking at the different types of the membrane proteins. But as you can see here, we're just looking at the composition of the biomembranes to see that apart from the lipids, which we did look at the last time, you can also have you can also have these membrane proteins, which are embedded inside of the cell membrane. Then we have different forms of them. Apart from the lipids, which we did look at the last time, here we are seeing another lipid, which we didn't look at, which is cholesterol. So cholesterol is also in there, in the cell membrane. Now, I think we got, because we didn't look at this cholesterol today, we will actually pay some attention to cholesterol and see, the functions of cholesterol as well, because I think that's the only lipid that we didn't look at when we're looking at the lipid membranes. Now, we want to also bring our mind to the fact that even though we're saying that biomembranes consist of these lipids and proteins and the complexes of carbohydrates with lipids and carbohydrates with proteins, all of these together. It isn't that if you take any cell membrane, if you take any cell membrane, the composition of it is going to be the same. Actually, you have biomembranes having different compositions, so you don't have a constant composition of biomembranes. These can depend on whether the cell is in a particular tissue or not. Even when you take cells of a particular tissue and you compare in different organisms, you'll find that you have some differences in the composition of these biomembranes that you are comparing. And so we have on the right hand side an example of cell membrane of rat hepatocyte. So that's a cell from the liver cells of a rat. That's in the cell membrane has been studied, and you could see that you have different compositions of lipids inside this cell membrane. The biomembranes of different organelles contain different compositions of lipids. And so we see, for instance, Golgi apparatus of that liver cell is having just about 10% of cholesterol. So if you look at the keys there, that violet there means it's cholesterol. And you can see on the horizontal axis, you have percentage. So it's from 0 to 100%. And we see cholesterol in the liver cell of the rat is, contains about just 10% of cholesterol. And you see about, say, 30% of phosphatidylcholine that is in the membrane of the Golgi apparatus. Now, you can do the same thing for different cells, different organelles, and do compare these things. So, for instance, even if you take the second here is inner mitochondrial membrane, and we should have also outer mitochondrial membrane. And if you do the comparison of the inner mitochondrial membrane and outer mitochondrial membrane, you realize the compositions are not the same. Even if you take the very first composition there, which is cholesterol for inner mitochondrial membrane, you realize it is a little bit less than what you see for the outer mitochondrial membrane. And you see the inner mitochondrial membrane also has more of cardiolipin in there than the outer mitochondrial membrane. And you see these kind of differences in the different biomembranes for the different organelles. Even if you take, for instance, endoplasmic reticulum, the last two, you see you have rough endoplasmic reticulum. So those ones will be having some ribosomes, attached on there. And you have smooth endoplasmic reticulum. And if you compare, you see that even in the rough endoplasmic reticulum, there is no cardiolipin at all, but you find cardiolipin in the smooth endoplasmic reticulum. And all the other components too, if you do look at them, you see that they all have different compositions. So I keep having this kind of message every time that we're having lectures that someone is outside a meeting and things like that. But the link I sent to you, everyone is able to join the class without any problem. You don't have to be outside anywhere. If you just click on the link, it will get you into the class directly. So I don't know what it is that is making people to say that they are outside and waiting for somebody to allow them to enter. You have been able to enter, so how come they are somewhere? All right. So you do see these kind of differences in the composition of the different membranes, depending on the organelles we are looking at. And even if you take a look at different cells, you'll see some of these differences. So clearly, biomembrane composition is not a constant thing. It differs from cell to cell. It differs from organelle to organelle. And so you see these kind of differences. This is not something which you should be scrambling in your head anyway, because I know many of you like to be memorizing all sorts of things. This is not something you should be memorizing. All right. Now, similarly, this brings us to what we refer to as asymmetry of biomembrane composition. What does it mean, asymmetry? So simply, if you take a cell membrane, we have seen that, say that if you take the biomembrane is a lipid bilayer. So you have on each side you have a monolayer. So if you look at the composition of the inner layer, for instance, and compare it to the outer layer, there too you don't have the same composition of the phospholipids that we are saying are the main components. So the lipids, which are the main components of the outer layer and the inner layer, are not the same. So that is what we refer to as asymmetry, in the sense that the biomembrane is not symmetrical. If you take off the let's say outer layer, it's not going to have the same thing as what you have in the inner layer. So that's what we refer to as the asymmetry of biomembrane composition. And here too, we're looking at an example of the red blood cell. That if you look at the composition of various phospholipids in there. For instance, if you take phosphatidylethanol you see in the inner layer, you see there's more of phosphatidylethanol in the inner layer than in the outer layer. On the other hand, phosphatidylcholine and sphingomyelins are more in the outer layer than they are in the inner layer. And you see that for the different lipids, like phosphatidylserine is having more of it. You are having more of it in the inner layer than in the outer layer. Same thing to do with the rest. phosphatidyl inositol, phosphatidyl inositol 4-phosphates, phosphatidyl inositol 4,5-biphosphate, phosphatidic acid. All of these, you find them in higher amounts in the inner layer of the biomembrane than the outer layer of that plasma membrane. So, I mean, that is what we refer to as asymmetry of biomembranes. Right. Now, if you take different cells from different organisms, here too you notice that there are differences in composition of components in them, still proving that cell membranes do not have constant composition. So, for instance, you take say in a nerve cell, take a nerve cell from humans. The myelin sheath of the nerve cell, has certain composition, proteins, phospholipids, sterol, different percentages. So here you see we're looking at some sterol, which is cholesterol in animals. And we did look at also these lipids, galactolipids, plasmalogens. These you find in the human myelin sheath. If you take, say, the liver cell of a mouse, for instance, you can see the compositions are not the same. Like protein is higher here than in humans. You see phospholipid composition is a little less than in humans. You see sterols there is more. And you notice that when you come down again, say for instance, I take a plant like a maize leaf. The proteins in there are even more, which is 47%. And you see phospholipids is lesser in there. You see sterols are much lesser in there, which is to say that you have less amount of lipids in the maize leaf, more amount of proteins in there, phospholipids a little bit less than what you find in mice. And you see these things throughout, that you have different compositions. Take for instance E. coli, the bacteria E. coli. The cell membrane is very high in protein. You can see that density of protein there is 75%, and it has just 25% of phospholipids, no sterols at all, and no other lipids. So it's mainly protein and some phospholipid together. So just to say some lipoprotein there, which is the composition of the cell membrane of E. coli. If you take this protozoa paramecium, for instance, it's also having a very high protein component, high component of phospholipids as well, and a less component of sterol, and it's having some specific type of sterol, stigma sterol. No other lipids in there. Now, like I said from the beginning, we did look at these lipids, like these galactolipids, plasmalogens, galactolipids, triacylglycerols, even forming, esters with glycerol. I mean, the fatty acids forming esters with glycerol. Of course, we didn't look at sterols, so obviously we didn't look at anything like the sterile esters as well. So today, as I said earlier, we will pay some attention to the sterols to add up to what we did with the lipids already. And then in the next lecture, then we'll move on and take a look at the proteins, like these membrane proteins, the different types that you can have. We can focus on that in the next lecture, looking at their the structures, the functions, we'll look at that. But for today, what we'll do is that we pay some attention to the sterols, and then we can close the chapter on the membrane lipids, so that the next time when we meet, we will then continue with the membrane proteins, and then yes, look at the structure, functions, yes, and then pick it up from there. Okay, so let's move on to the sterols. Now, the major sterol that you do talk about a lot is cholesterol. Cholesterol we've seen to be also a major component of biomembranes, and you should pay attention to the fact that cholesterol is found only in animal cell membranes. There are other types of sterols, as we saw in the table before, that there was some maize leaf that was having some sterol in it, but we'll see what kind of sterol do we find in plants. But in animal cells, what you find is cholesterol in animal cell membranes. And you take a look at the structure, you see that, just like you have these fatty acids with the carboxylic acid group, which is hydrophilic. Then you have the hydrocarbon chain, which is the hydrophobic tail. You see that cholesterol is having some hydrophilic head as well, which is a polar head. You see there, the hydroxyl group on carbon number 3. And the general structure of cholesterol is such that you have these 4 rings. So you want to pay attention to that all cholesterols will be having what we are referring to as the steroid nucleus. So you have these 4 rings fused together, the 6-sided rings fused together with one 5-sided ring. And you see that there is some side chain, which is a straight chain hydrocarbon there, which is on that last the D ring, the ring which is labeled D, the 5-sided ring. You see that there is some side chain which is on it. This straight chain residue can be different for different steroids or different sterols. So you will see that it is actually this portion, this straight chain portion, which differs in different sterols, but the steroid nucleus is constant. You see the same steroid nucleus for all of them. So it is only this side chain which will be different for the different sterols. And the function of these sterols, even though we'll come to look at it much more, is to regulate membrane fluidity. As you can see, if you compare this structure to the structure of the fatty acids, you see that the fatty acid is a straight chain structure. Some of it will be saturated, some of it unsaturated, and we did look at how that is related to melting point, depending on the number of carbons or the molecular mass of the fatty acid. At the same time, we also then looked at how the saturation or unsaturation, that is the number of double bonds which are present in the fatty acid, how that relates to melting point temperature and all of that. But here you see that cholesterol is having a ring structure, and even it is 4 rings fused together. I mean, that should tell you that this structure is going to be much more rigid to be broken down, which means that the melting point for this will be much higher, compared to any of those fatty acids. By virtue of these rings, because these rings require more energy to be broken down. And so you will see, as we continue, we'll see how this also affects, say, melting points, temperature, and then we'll relate it to how that can regulate stability of the biomembrane. Right. Now, as I was saying, cholesterol is a sterol that you find in animal cells, but in plant cells you have different types of sterols that you find. And so here we're looking at some different examples. These are not all of them anyway, but I mean, just to take a look at the fact that yes, the sterols are having similar structure, having the same ring, what we just saw as the steroid nucleus, the 6-sided rings, 3 of them, and then 1 5-sided ring, all fused together. And then you have the side chain, which is on that 5-sided ring, which is different in all of them. So if you pay attention to that side chain, you realize there are differences. Even if you take a look at, say, beta cetosterol and compare it to the one below it, the stigmasterol, you see stigmasterol is, I mean, quickly you can even see stigmasterol is having 1 double bond here, which you don't see in the beta cetosterol. So that's a difference. And even if you come to look at campisterol, it's also looking different. It's not the same as any of these. Similar thing with brassicasterol. It's also different from all the others. So you see that even though you have the steroid nucleus being constant in all of them, you have these differences of the side chain, which is on that ring, the D ring over there. Apart from even these sterols, which you find, I mean, what we say phytosterols in plants, there are some structures which you also see related to this, which are referred to as stanols. So stanols differ from sterols in the sense that the nucleus looks the same. I mean, the steroid nucleus, except that if you pay attention, you see that the second ring here has 1 double bond in it. For all the sterols, you see the second ring is having double bond in it there. Campisterol is having that double bond. Brassicasterol is having that double bond. Stigmasterol is having that double bond. Cetosterol is also having that double bond. But when you take the stanols don't have that. So you when you read biochemistry books, you may see some of those as well. They are not sterols, they are stanols, but they are related. They carry out similar functions. Now you see another thing that you should also bring your mind to, much more practically, is that you see we do extraction from plants. A lot of you do your project works, you do some of these extractions from plant leaves, maybe plant roots, and things like that. And then you use some of those extracts testing for some biological activity. Those of you who are interested in doing some of these macrobiological research works, you do test activity of some of these extracts against some bacterial strains and things like that. You see, when you do some of these extractions, you ask yourself, what is it that you are extracting, particularly if what you are extracting is lipid. Some of these lipids that you are extracting, apart from the fact that many of those lipids will be some phospholipids, which are coming from the cell membranes, you also have some of these sterols, which are also being extracted from the plant's biomembranes. So you generally say that you are doing a plant extract. But if you should ask yourself, where exactly of the plant? In any case, that plant you say you are doing extraction from, the plant is made up of what's the basic unit of life? It's a cell. Now, if you take the cell, which specific part of it are you getting the extract from? And you realize you are doing a lot of the extraction from the biomembrane. Now, this brings us to also a discussion on the use of these sterols as some supplements in place of cholesterol. So, in fact, these sterols have been applied for conditions of that are having increase in cholesterol in say blood, so that there are supplements which mainly consist of these phytosterols that are prescribed, that are taken to reduce cholesterol levels in blood. And even though some of these things are still continue to be studied, not all of them have been fully, been confirmed to be say biologically active for dealing with problems like cardiac issues, high cholesterol levels in blood and things like that. But there are some medicines out there on the market that contain these things, contain some of these phytosterols in them. I mean, that is because they are sterols that are coming from plants. They can easily solubilize cholesterol, and so they don't get accumulated in blood vessels, forming plaques and causing increase in blood pressure and other cardiac conditions as well. So I mean, this it's a practical application of these things, and so that the next time you say you are doing some extract, specifically that in fact what you are extracting is coming from the cell membranes, and it's the structure of it. If you look at the chemical structure of it, is similar to that of the cholesterol. Now we can move on and speak much more about how the cholesterol and let's say generally sterols, because we are not speaking only of only the animal biomembranes, but can also be in plant membranes, because as we've seen, you have different types of sterols in addition to cholesterol, which you find in animal cells. So the sterol composition of biomembranes. Now, if you take a close look at the structure here, that is looking at let's say a monolayer of biomembrane, where you have phospholipids with the polar hydrophilic head and then the non-polar hydrophobic tails. And now you do remember when we were looking at these phospholipids with some fatty acids with double bonds in them. Those double bonds result into some bend in the structure of the hydrophobic tail, because we did say that naturally these fatty acids are in their cis configuration, and in their cis configuration you have steric hindrance or steric repulsion being higher, which results into a bend in the structure, and such a bend then results into some instability in the positions where they are in the cell membrane. You do remember that. Here we are not showing any of such double bond. We're just showing, say, cell membrane with a hydrophobic tail, and then you have some cholesterol which is embedded in between these phospholipids. Now, what do you notice? You see, you have this the ring structure. I mean, the steroid nucleus, which is occupying such space, position inside, in between these fatty acid, these fatty acid layers, and you also have that side chain, which we saw to be different in different sterols. Now, if you see this structure, you can clearly see that this is a more compact structure, even though you see it's like a bigger space between the phospholipids. It is a bigger space because of the structure of the steroid. The steroid is a much bigger molecule and it can occupy such a space between these fatty acids. Now, in order to understand how this can result into some effects on the stability of the membrane, we have to take a look at how the steroid behaves in terms of how to say the transition temperature. Or let's talk about, say, the melting point, because we did see how melting point of these fatty acids was related to their structure, looking at the molecular weight, that's the number of carbons, and also the number of double bonds which are present in the molecule. Now we see that when you take a cell membrane, the cell membrane can exist usually, optimally at a particular temperature, what we are showing here as a midpoint temperature, which is also what we refer to as a transition temperature. Now, transition in the sense that you see you have a membrane that at a particular temperature, I mean the midpoint, what you have there, Tm, which is the midpoint or the transition temperature. From at this temperature, the membrane is more stable, so that if the temperature increases above this, then you have the membrane becoming more and more fluid, because of course the lipid molecules in the lipid molecules in the membrane are going to be more agitated with increase in temperature, and so you have the membrane getting more fluid. The opposite of what can happen is that if that temperature is reduced to at a lower temperature, you have the lipid assuming a more solid character. That is, the membrane will be getting solidified. But you also see you have a relation also to do with what we were just referring to as saturation of the fatty acids, or the molecular mass. That is when you have the number of carbons increasing. What can happen to these things? Now, you do see that when you have an increase in the fatty acids, with more carbons in it, or you have more saturated, like that is single bonds in it. It means that the amount of energy or the temperature which is required to move that membrane into a fluid state will be higher. You need more energy to agitate, saturated fatty acids or bigger fatty acids in order for that membrane to become more fluid. I mean, contrary to that, there will be less amount of energy which will be required in order to make a lipid or the biomembrane become, more fluid or more assume a more solid character. Now, what you notice here is that you see for shorter or more unsaturated fatty acids, you have a lesser amount of energy in there, which is required actually to actually make that molecule more fluid. So you see that if you do compare sterols, which will be in the structure of the biomembrane, and you see that the sterol is having rings, 4 rings, which will require more energy in order for that membrane to be moving between these solid, states to a fluid state. Now, what sterols come in here to do in terms of stabilization of the membrane is that, as you see, you have the region around the midpoint, that is the transition temperature. You have that region extended by the sterol. So it means that you can have the membrane existing at a stable state with sterols, with more sterols in it, so that more energy is required in order to move the membrane from that midpoint into a more fluid state, or more energy is also required, or just to say a freezing energy is more freezing energy is required in order to move the membrane also to, say, a solid state. So in that case, what it means is that the more sterols that you have in a membrane, even when the membrane is at a lower temperature, than usual, the membrane can still withstand that lower temperature and not become solid. The same way, it will also withstand a higher temperature, so that if in a normal case at a particular high temperature, without sterols, the membrane would have reached a stage of destruction, but with sterols, the membrane will still be stable at such a high temperature. So you see, sterols play such a key role in stabilization of the cell membrane at different temperatures, in different conditions, whether the temperature goes low or the temperature goes high. You see that the sterols are playing such a role of stabilizing the cell membrane in this sense. See, by widening that gap of stability around the transition temperature. Right. This brings us to, simple conclusion that there are factors that affect membrane fluidity, which can be temperature, as we've just seen, and even from our last lecture, degree of saturation of the fatty acids, how much of double bonds are in there or no double bonds, will also affect membrane fluidity. Then lastly, what we have just seen is that the sterol component of membranes can also affect the fluidity of the membranes. So you have these three major factors that can affect membrane fluidity. Right, now we can also extend this to take a look at, say, let's take say bacteria E. coli, which had been studied. Look at a composition of various fatty acids in the membrane. Compare that to the conditions under which these bacteria had been cultured. So this is to see how organisms actually adapt to different conditions. In terms of what we've just looked at, looking at those factors that affect membrane fluidity. Now, this is some experimental work which had been done with the bacteria E. coli, which had been cultured at different temperatures: 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, and 40 degrees Celsius. And then the components, the fatty acid component or the lipid component of the membranes of these bacteria were then compared to see the differences of these bacteria. Now you see that it's the same organism, E. coli, except that it is being cultured at different temperatures. And you see the bacteria adapts such that you see that at a lower temperature of 10 degrees Celsius. So apart from looking at the compositions of the various lipids like the myristic acid, palmitic acid, palmitoleic acid, oleic acid, hydroxymyristic acid, let's bring our focus to the last thing, which is the ratio of unsaturated to saturated fatty acids, because that is where actually the story is. To say that if you look at the biomembrane composition of E. coli that was cultured at 10 degrees Celsius, you see that there is a high ratio of unsaturated to saturated fatty acids. In other words, you have a high amount of unsaturated fatty acids when the bacteria was cultured at 10 degrees Celsius. Why that? Unsaturated fatty acids, if you have more of them in biomembrane, what does it mean? It means that the biomembrane should be more fluid. And you see, that is to compensate for the low temperature of 10 degrees Celsius. Because the low temperature should be resulting in less agitation, less energy for the movement of the lipids. So that if there is less amount of energy for movement of lipids, that will mean that there will be less fluidity. So to compensate for that less fluidity, the bacteria, its cell membrane in growing, grows with more unsaturated fatty acids to compensate for that fluidity. And you see the trend is that as the temperature increases, the ratio of unsaturated fatty acids to saturated fatty acids reduces. And so you see, at a temperature of 40 degrees Celsius, same E. coli, which is cultured at such a temperature, is having unsaturated to saturated fatty acid ratio being 0.38. In other words, less amount of unsaturated fatty acids were found in the membrane, or more saturated fatty acids. Because if you have more saturated fatty acids, it will mean that more energy will be required in order to make that fatty acid fluid. Already the temperature, 40 degrees Celsius, is high enough, to agitate the lipid molecules, making it more fluid. And so there will not be the need to be having more amount of unsaturated fatty acids there. To compensate for that, the bacteria will rather need more saturated fatty acids in the membrane. And so you see that trend that as the temperature at which you have the culturing of these bacteria was increasing, then you have also the increase in unsaturated fatty acids there. That's an interesting thing to see, that you actually have organisms adapting in such a way that depending on the temperature in which they are growing, they will have particular compositions of these fatty acids in them, being it either saturated or unsaturated. And even if you take a look at, say, the number of carbons, that is to say, increase in, say, molecular mass of the fatty acids that are in these membranes. Perhaps you can notice that, for instance, in this fatty acid, which has maybe 18 carbons, oleic acid, for instance, you can see what's the trend? The trend is that it's also reducing as the temperature increases. You have that fatty acid decreasing there. It's a saturated fatty. Okay. All of them are unsaturated, because 18:1 is an unsaturated fatty acid. So as the temperature increases, you have some reduction of that. But interestingly, you have palmitic acid, which is having no double bond in it, that is saturated. You see that it is in high amounts for the E. coli that was cultured at 40 degrees Celsius. It's having a high amount of this palmitic acid in there. That is a high amount of saturated fatty acid to compensate for that high temperature there. All right. Now, and actually you can see that it is along that palmitic acid that you see most of the change, because you can see that 18 for 10 degrees Celsius, and then for 20 and 30 degrees, here not so much of a difference, not change, but 40 degrees and the difference is big. You see that palmitic acid is in a very high amount there. So you can, and the same way, even if you take myristic, you also see that it increases for the E. coli that was grown at 40 degrees Celsius. So you can see these kind of trends and begin to see that the organism has actually adapted to these temperatures with the amount of saturated and unsaturated fatty acids that have been synthesized by the organism to be components of its cell membrane. Now we can move on and talk about what we refer to as membrane dynamics, particularly going into even movements of these components of the cell membrane, the lipids. But before then, with what we had just talked about in the last slide, you see that you can have different states of the membrane. You can have membrane in a form of, say, a gel-like membrane, which is sort of semi-solid, and that's going to happen at lower temperatures. Logically, if you have a lower temperature, there will be less movement of the lipids in the lipid bilayer. So if there's less movement, you have such a gel-like structure, what is being shown at the top as a paracrystalline state, the gel-like structure. So you actually have less movement, and so you can see that the phospholipids are actually distinct. You can see them clearly. But then as temperature increases, you have some liquid ordered states. That is to say, at an intermediate temperature, at a temperature not so high, you have slight movements, and so you have some some liquid states, not gel, but ordered. And as the temperature increases, the more you have a more fluid state of the membrane, what is being referred to as a liquid disordered state. And that's what you actually see below here, the fluid state, where you see it, you cannot see distinctly the phospholipids that are forming the bilayer. And so you see that heat, or less temperature, increase in temperature results in more agitation of these phospholipids. And so the movement of it, of these phospholipids, is what you're referring to as some thermal motion of these phospholipids, which then results into a transition from a gel-like structure to a fluid structure. All right, so I think some of these things are logical and straightforward that you can get them. Now we move on to look at membrane movements. Because you say the membrane is fluid, it moves. So if the membrane is fluid and it moves, what types of movements do we have in the membrane? Now we have two major kinds of movement in the membranes, two major kinds. You can have uncatalyzed movements. So uncatalyzed in the sense that there is no participation of an enzyme in the movement of the lipids. Those phospholipids which are in the bilayer, they move themselves without any participation of enzymes. So those are uncatalyzed movements, and you have two of them. You have what you refer to as a transbilayer diffusion, also referred to as flip-flop. You have lateral diffusion, and then you have catalyzed movements, which are movements that are aided by the activity of enzymes. And even in some of them, you have the expense of energy in the form of ATP. So you have some ATP-dependent flip movement, ATP-dependent flop movement, and you have one of them which is ATP independent. So energy is not used in this case. Even though it's a catalyzed movement, there is no use of energy in that, which is flip-flop. So this last one is like a catalyzed flip-flop of the first one, which is uncatalyzed. Now let's take a look at these things pictorially. Here is just a list, and then we take them one by one and begin to look at them so that you will have a better understanding of what we are referring to as these kinds of movements. The first one is the transbilayer diffusion. Transbilayer means that the movement of the lipid is from one of the monolayer to the other monolayer. So it's transbilayer. It's diffusion. So diffusion in the sense that it's uncatalyzed. There's no use of energy. There's no participation of an enzyme in here. And this movement is referred to as flip-flop. So if you take the cell membrane, it is either the movement is from the outer membrane to the inner membrane, or it is from the inner membrane to the outer membrane. Both of them are transbilayer. If it is into the cell, that is from outside into the cell, it is referred to as flip. If it is from inside of the cell to the outside, it is flop. How to remember well is I is in. O is out. So if you say flip, it means the movement is from outside monolayer to the inside monolayer. I mean, if you take the cell, the cell is having an outer extracellular side and then the inner side, which is the cytoplasm. So if the movement is from the outer layer to the inner layer, the movement is flip. If it is from the outer layer, if it's from the inner layer to the outer layer, it is flop. Now, but you do refer to both of them as flip-flop. There is no use of energy, no participation of any enzyme in this type of movement. And this movement is a very slow one. With a half-life estimation of say in days, this can be occurring. I mean, it's very slow. That is not a fast movement. And this is, we say that the membrane is fluid, so movements occurring in different ways. And so such kind of movement of a lipid from outside to the inside can occur, but it's very slow. This can take a number of days to occur, say maybe 2 days, 3 days, and then there's like a full movement of the phospholipid from one layer to the other layer. So that is one of the uncatalyzed. Now the second catalyzed is what we are referring to as a lateral diffusion. Now, even though the arrow is drawn to the right-hand side, it can be to the left-hand side as well. Because like you have a membrane in which you have, some phospholipid which can move in the same monolayer. This time around, it's not moving from one of the monolayer to the other. That's why we're referring to it as lateral. It's not transbilayer, but it is lateral. It's lateral diffusion. And these two, of course, is within the same monolayer. So it's fast. It's a very fast movement which occurs. In fact, the membrane, as fluid as it can be, you have lots of these lipids which can easily be moving in the monolayer, and so you have such lateral diffusion also occurring, and you can see this is very fast. The movement can be 1 micromolar per second, not so much of a big distance, but little movements. You have such movements occurring, which are 1 micrometer is not a big distance per se per second. So you have that lateral diffusion. All right. Now we move to the catalyzed movement. So this is where you have some enzymes that will be acting in the process, and you have here. You see, we did refer to the movement from the outside to the inside to be a flip movement. Now, in the catalyzed flip movement, you have the enzyme flipase. And remember that this is occurring with the expense of energy in the form of ATP. So that is an ATP dependent movement. So that you have the movement of phospholipid from an outer layer to the inner layer. And that is catalyzed by the enzyme flipase with the use of energy in the form of ATP. The opposite of that, you have the movement of a phospholipid from the inner layer to the outer layer, which is catalyzed by the enzyme floppase. Here too, you have expense of energy in the form of ATP. Now, there's one enzyme, scramblase, which can do both. It can catalyze both flip and flop. But with this enzyme, there is not going to be the expense of energy, which means the movement, which is catalyzed by scramblase, is an ATP independent movement. So you see these differences. And then you take note of the enzymes, flipase, flopase, scramblase. And you also, clearly, you see this is the outside of the cell that is above, to show that outside of the cell above and inside of the cell below. To show that, yes, if you have a movement from outside to the inside, that is a flip. So flipase. And also if it's from inside to the outside, that is a flop. And so the enzyme there is flopase. So yes, these movements are also transmembrane movements, but they are catalyzed. There are enzymes. In other words, these enzymes they are embedded proteins in the biomembrane, which catalyze these movements. All right. Now, another kind of movement which we can talk about is what is referred to as Hop diffusion. And what does it mean? Hop diffusion. So, studies have shown that, if you take, let's say, a particular phospholipid and microscopically study its movement to see how this phospholipid is moving to which directions, to which areas, how does this phospholipid move about in, say, a monolayer, for instance, a monolayer, say the lateral diffusion. And what has been seen with different phospholipids is that the phospholipids actually do not move too far away. It's not so much of random area of movement. So for instance, you take let's say the yellow movement lines which are shown, you realize that phospholipid is moving around a particular region throughout, and then at a point it makes some occasional hop to a different region. And so that is what is being referred to as hop diffusion. But you can see that its movement is within a particular region, which brings to what is referred to as molecular fences, to say that if you take a particular phospholipid, it will be moving within a particular region and will not go beyond that. If you look at all of them, like if you take the region which is green, that phospholipid is also moving mostly around a particular region and doesn't go beyond that. The blue, the violet, the same, they are all moving within a particular region and not going beyond that region, except that they move at one particular place for a long time. And so you see that all those places are very covered with no spaces, and then occasionally they hop to a different region and then they can come back. So you have such a kind of a movement which is traced out, and you can see it clearly that they are moving within a particular region, mostly for a long time. And so the conclusion that you have some kind of, theoretically, you have what is being referred to as molecular fences for these phospholipids that are moving. So that is what is being referred to as hop diffusion. Now, to bring this chapter of lipids to a close before we move on to look at proteins, the biomembrane proteins in our last lecture. Let's end this on a more real life, practical, issue, practical cases, which is, we did talk about the fact that the membrane is dynamic. It's not static. And also in the sense that you have these lipids undergo recycling. So that one of the things we didn't see was that even though we did talk about lipid metabolism last semester, looking at in addition to all the other metabolic processes that we saw, we have these membrane lipids also undergoing some degradation and going through a process of recycling. Perhaps we did see phospholipases, how they break down phospholipids, to say that phospholipase A breaks down the first ester bond between a fatty acid and a glycerol backbone. Phospholipase A2 breaks down that ester bond between the second fatty acid and the glycerol. Then you have, so for phospholipids, you have a phosphoester bond that forms between the glycerol backbone and a phosphate group. And the enzyme that breaks that down is phospholipase C. And you have phospholipase D, which breaks down the phosphoester bond between the phosphate group on the glycerol backbone and a side chain, which is on that phosphate group. We did see that for these phospholipids, you can have different head groups, not specifically what you are seeing here, which is some inositol. No, that's not the only thing. We did see even some amino acid can be there. You can have different carbohydrates or even some lipids. You can have them there. So the only thing is that this particular bond, the phosphoester bond, which is between the head group and the phosphate group, which is on the glycerol backbone, that enzyme that breaks that bond is the phospholipase D. So which means that all these components, after they are broken off the glycerol backbone, can go into the formation of other phospholipids. So that is what is being referred to as the recycling pathway of these lipids. And this occurs throughout. I mean, regularly you have this thing, or it's happening constantly with the membrane lipids. Now, this brings us to some disease conditions that can occur due to some genetic disorders that results into some abnormal accumulation of the membrane lipids. So we did see some of these, ceramide lipids having the backbone sphingolipids, onto which you have, other side chains that are other head groups that are bonded on these ceramides. And we did see these things, a lot of them in, say we said you mostly find them in some nerve cells. Now, what happens is that, there are some genetic diseases that are inherited from parents. A lot of times, these parents are carriers. So if you have two parents that are carriers, and then they give birth, and sometimes it does happen that maybe in about 25% of the children, have the risk of developing some of these genetic disorders where there is lack of or deficiency in some particular enzymes that are required in the metabolism of these membrane lipids. So we have, say, a membrane lipid, which is GM1 in these nerve cells, that you have the enzyme, the beta galactosidase. I think we've met beta galactosidase a couple of times in the metabolism of lactose, breaking lactose down into galactose and glucose. But in this case, what beta galactosidase is doing here is that it is breaking off that terminal galactose residue, converting that membrane lipid GM1 to GM2. So that is just the beginning of a process. So different reactions will occur, and you have the lipid breaking down to different components until the whole recycling process goes on and on. And you have, if you have this beta galactosidase deficiency for such persons, which is a genetic disorder, then what it means is that you will not have this reaction, which is the breaking off of the galactose, that terminal galactose residue, which also means that there's going to be an accumulation of this lipid membrane, the membrane lipid GM1 in the system. And a lot of times, if you have GM1 being accumulated in the nervous system, and then it results into what is being referred to as generalized gangliosidosis. Some of these results into some mental disorders, some mental retardation, some growth abnormalities. You do have such happening. Similarly, you can have what is referred to as Tay-Sachs disease, which also results from, say, deficiency in the enzyme hexosaminidase A, which is involved in that second reaction where you have the breakdown of this cleaving off of that glycosidic bond between that terminal N-acetylgalactosamine residue with the galactose residue in there. Because you see in the reaction that terminal N-acetylgalactosamine is cleaved off. And so if you have hexosaminidase A, which is deficient, then what it results in is that there's going to be accumulation of this membrane lipid, which is GM2, and that will result into that condition of Tay-Sachs disease, which is also one of these rare genetic conditions that affects, say, hearing and sight of children, which are born with this condition. Now, you can also have other conditions like, say, maybe Gaucher's disease, also resulting from, say, deficiency in this enzyme, glucocerebrosidase, which, of course, then will be resulting in the accumulation of this glycosylceramide. You have another one here where you have the enzyme sphingomyelinase, which breaks the bond between the ceramide backbone and phosphocholine in sphingomyelin to form the ceramide. That's going to be the sphingosine with a phosphate group to have the recycling to form another sphingolipid. And if you have a deficiency of sphingomyelinase, it results into another genetic disorder, the Niemann-Pick disease. And so you see that many of these deficiencies of these enzymes, many of these genetic disorders are also linked to metabolism of membrane lipids, which can have some central nervous system disorders, some serious neurodegenerative disorders for different persons. Some mental health disorders, some depressive disorders, things like that. So these are some very real life practical things which can happen with some of these things which you are studying with the membranes and the types of lipids that are in them. And if you relate it to metabolism with specific enzymes, and these enzymes are proteins, which means that those proteins will have to be synthesized from the genetic code that is in the DNA, so that if there is some kind of mutation, some change in the genetic code in the DNA, that can affect the synthesis of the protein. And that protein may become inactive, or perhaps the synthesis of the enzyme is deficient in such a way that not enough. And all of these can be resulting into some of these conditions. Of course, these are not conditions that are so common among people, but if you go into some populations, if you go into the epidemiology of it, you may find that in some specific populations, you have maybe one in a certain number, 1 in 1,000, 1 in 300, 1 in 500 people who develop some of these diseases. Depending on the population in which you go, you find these things which are occurring in different populations. All right, I think I'll end it here, and then I open the floor. If you have any questions, you have any contributions, the floor is open. You can write in the chat and I will respond, or you can equally raise your hand. I will enable your microphone, and then you can unmute yourself and you speak. The floor is open.